Okay, this is a brief overview of photosynthesis. Make sure that you are pausing the video as needed to take notes. So photosynthesis is a process that autotrophs use in order to take the energy from sunlight and convert carbon dioxide into chemical energy that's stored in the form of carbohydrates. This is an equation that you've likely seen before, showing that photosynthesis is a conversion of carbon dioxide, 6 molecules of that, with 6 molecules of water into 1 molecule of glucose and 6 molecules of oxygen. And as we'll soon learn, photosynthesis and cellular respiration are intimately connected. In fact, these two equations are simply the same as one another, but backwards. Just a little review. You might not need to write this down. Remember that we can classify organisms based on their use and acquisition of energy. Autotrophs will use sunlight or chemicals directly to produce food from simple inorganic molecules, including plants, algae, and some bacteria. Heterotrophs get their energy from the food they consume, like animals, fungi, and some bacteria. But ultimately, all life depends on autotrophs. Be sure to get this diagram in your notes and follow along as I explain. Because it's a cycle, you can start anywhere, but we'll start right here. Light energy is captured by autotrophs, and then they will capture carbon dioxide and water and convert these molecules into organic compounds and oxygen through the process of photosynthesis. These organic compounds and oxygen will then be used by both autotrophs and heterotrophs through the process of cellular respiration for energy, but in the meantime, they will release carbon dioxide and water as waste products. These waste products are then going to be used again by autotrophs during photosynthesis. Both photosynthesis and cellular respiration are biochemical pathways. So these are chemical pathways that occur in living things. So again, pause the video so you can make sure you get that diagram into your notes. As you'll see in this presentation, there are a few requirements that photosynthesis requires. Chloroplasts are needed by plants and algae. However, there are bacteria that do photosynthesis, and as you may recall, they do not have organelles, so they don't have chloroplasts, but they still have all the molecules to carry out the same process. Sunlight is required. Pigments within leaves. And again, if you're not a plant and you don't have leaves, algae do have leaf-like structures. Bacteria don't have leaves, but they still have pigments. Water, of course. And carbon dioxide, which is the building block of those organic molecules like glucose that end up getting made. So we're going to focus in on plants and algae and leaves. If you were to look at a leaf and magnify a small section of that leaf until you saw a plant cell, you'd see these green structures that are called chloroplasts. If we zoom in on them, there are these double membraned green structures that have these little stacks of flattened green discs called thylakoids. They kind of look like green pancakes. Each thylakoid has a space within it, and then the entire interior of the chloroplast is filled with a solution called stroma. If we were to then zoom in on one of these thylakoid membranes, we would see what looks like a cell membrane, but then embedded proteins, and this is where plants and their pigments have the ability to actually split water. And that's a really kind of unique process, and it's why the plants actually need water. Thylakoids then are these interconnected flattened sacs within chloroplasts with pigments like chlorophyll, and they're embedded within its membrane. So we're going to focus on sunlight here for a moment, and sunlight sends out packets of energy that we call photons. Photons have different amounts of energy depending on their wavelength. And all of these different types of energies that come from the sun are all part of what we call the electromagnetic spectrum, or EMS. It's a range of all forms of light from low energy to high energy, from radio waves to gamma waves. And so if you look at this spectrum, AM radio waves are 10 to the 4th meters. And so these are really, really long. Then you get into shortwave radio waves, TV waves, FM, radio. Infrared is not something that we can see, but some animals can. Ultraviolet rays, think getting sunburns. X-rays, which can penetrate our bodies, right? But they bounce off of bones. And then gamma rays, which can penetrate all parts of our body. These have very tiny wavelengths, so this is why they're very powerful. They go quickly through tissues. There's a narrow part of this spectrum called the visible light spectrum, and this is everything that we can see. White light that comes from the sun, if passed through a prism, emits these different wavelengths that we perceive as the colors of the rainbow. Red colors have the longest wavelength and the lowest amount of energy. Purple colors have the shortest wavelength and the highest amount of energy. But we're talking really tiny wavelengths in nanometers, and a nanometer is 10 to the 9th meters, negative 9th. So this visible spectrum is that small range of light that can be perceived by organisms. Roy G. Biv is red, orange, yellow, green, blue, indigo, and violet. And so here you see, you probably recognize this from a famous band's album cover, hopefully, white light coming in through a prism and coming out being split into its respective wavelengths of colors. So the colors that we see, this is kind of interesting, are actually just what's reflected back to us. So when you see an object that's red, like a red apple, what's happening is the pigments in the apple are actually absorbing all of the colors except for that red that you see. And the red is being reflected back at you. So you might ask the question, is the apple red or is it not red? And in totality, white light is all colors being reflected back at you, whereas black is where all the colors are being absorbed. And so think about what color of a t-shirt you would rather wear on a really hot, bright, sunny day if you wanted to stay cool. So we mentioned earlier that pigments help to capture sunlight energy. So these are actually made of protein, and they have the ability to capture the energy from the sun. These are embedded within the thylakoid membrane, remember those little green pancakes, and they can absorb or reflect certain wavelengths of light. Plants have multiple types of pigments, the main pigment being chlorophyll A. If you look at the graph below, chlorophyll A is this red line, and underneath you can see the wavelengths of light, the colors of the rainbow, that it absorbs. So the percentage of light absorbed is on the y-axis. So you can see it's really good at absorbing some violets, not so good in the middle, but then again over in the reds. So that means that they're reflecting everything in between, and they tend to appear a bluish green color. This is the main leaf color that you will notice. But plants don't stop there. They have the ability to capture pigments from all kinds of wavelengths of light. And they do this by using accessory pigments. This helps to maximize the energy that they get from the sun. Chlorophyll B, denoted by the yellow line down here, you'll see that it shifts more toward the center where it can absorb some of the blues and more of the reds and even some of the oranges from the sun. And so these leaves will appear a lighter green than those that have a dominant chlorophyll A. Carotenoids, denoted by the purple line here, they only absorb in here in the blue and the green, but they tend to appear yellow-orange to us. Truly, think carrots. And there are leaves sometimes that also have that kind of same orangish color or yellow color. You've probably seen leaves in that range. And not on this graph are xanthophylls, which absorb violet, and they tend to appear red. So think about this idea that plants can have differently colored leaves, and we'll mention that they actually will have different colors of leaves at different seasons of the year. Okay, let's get into the nitty-gritty of photosynthesis. So there are a couple of stages of the reactions that occur, and in a couple slides, there's a big drawing that I'm going to have you put down in your notes, but for now, just write down what you see in green. So they're light reactions, part one. These are known as the light-dependent reactions. That's because they require light. So if you notice the picture, we're inside of the chloroplast, inside of the thylakoid, looking specifically in the membrane of the thylakoid. So this step here is all about capturing solar energy. That's very important that you note that. And you'll see that the light, the photon, is coming in and striking this pigment. So the pigments have names. They're called photosystems 1 and 2, and that's not crucial that you know, but it might be kind of more filling out the story for you. So this is happening in the thylakoid membrane, and the light is absorbed by chlorophyll. And the chlorophyll everything is made of atoms. Atoms contain electrons. This light will then excite the electron to a higher energy state. And the light energy and its excited electrons is then used, this is pretty amazing, to split water. It literally splits water into oxygen, which is oxygen gas, and for hydrogen ions or protons. And the electrons get excited and they're going to be passed along this electron transport chain, which we'll look at on the next slide. But what's also important is that right here, this oxygen that's generated, this is that oxygen that we breathe in the atmosphere. And so this is released through structures called stomata, which we'll talk about, out into the atmosphere. This is where that comes from in step 1. Step 2, this is the rest of the electron transport chain. And so I would think of this as being more like charging the batteries, making what we call ATP and NADPH. This also happens in the thylakoid membrane. So as the electrons are excited, they're used to do a couple of important tasks. They are pumping these hydrogen ions into the center of the thylakoid, creating a concentration of hydrogen ions that is higher than outside. The second pigment, the photosystem here, is going to absorb more photons. And in this case, it's going to combine this hydrogen ion with an electron and generate what we call an electron carrier. This NADP plus a hydrogen ion will form NADPH. Now, this is a fancy molecule that you don't really have to know much about other than it's an electron acceptor and it carries energy. You can think of it as being like a charged battery. So this is that rechargeable battery that you're going to plug back in and charge up. So this is one of those now charged batteries, and it stands for nicotinamide adenine diphosphate, but you don't have to know that. Additionally, what happens here is this is a really important enzyme that's called ATP synthase. And as these hydrogen ions are trying to fight their way back out, think about being in a crowded space and people are trying to funnel out through an exit. They are flowing through ATP synthase like water out of a hose. That generates a lot of kinetic energy. And what's not shown here is the end of this ATP synthase actually spins like a wheel. And that energy helps to bring together this discharged battery or used battery of ADP, adenosine diphosphate, and a phosphate molecule to generate the most important energy molecule in cells called ATP or adenosine triphosphate. These two molecules, ATP and NADPH, are then going to be used in the next stage, the Calvin cycle, to make glucose or sugars. So that's a deeper explanation than what you need to know. But what you do need to know is the light reactions is this idea of kind of charging the batteries to make these two energetic molecules of NADPH and ATP, which will be used in the next stage. So step 3 is the dark reactions, or the Calvin cycle, also known as the light-independent reactions. These don't necessarily happen in the dark, but it doesn't need the light. And so that's why it's called the dark reactions. And they're also known as the Calvin cycle because of the scientist who first described this process. The dark reactions happen in the stroma of the chloroplast. So remember, inside the chloroplast, you had all those stacks of thylakoids. They're bathed in a syrup that we can call the stroma. And in order for this to proceed, we need carbon dioxide, which is the building block of glucose. So meanwhile, carbon dioxide would have diffused into the stroma from the atmosphere through little pores called stomata. Don't get stroma and stomata confused. Stomata are openings. Stroma is the syrup. So this process is really important. It's going to make glucose, C6H12O6, from the carbon dioxide with the help of those energetic molecules of ATP and NADPH. This is a repeating cycle. Again, you're writing down what you see in green. You can add more if you like. Carbon dioxide will enter through the stomata and wind up into the stroma. And through this process, this circular process, this carbon dioxide is ultimately converted into molecules, organic molecules like carbohydrates. There are bits and pieces of the cycle that you don't need to know, but I like to show this so you can see that it's a multi-step process where we start off with 3 one-carbon compounds with the help of an enzyme called rubisco. We join multiple carbon dioxides together to form something called PGA. We make a couple of them. And then here we're going to use the ATP and the NADPH to turn these into a different kind of carbon compound, PGAL. And then PGAL will be joined together with additional PGALs to form glucose molecules. But then what's left in the cycle will be recycled to stay here and keep helping the cycle proceed. So we're going to need more ATP to convert the PGAL into ribulose bisphosphate, or RUBP, which is a 6-carbon compound which will re-enter the cycle. Again, you don't need to know all those details at all. You just need to know that those energetic molecules of ATP and NADPH are used to convert carbon dioxide into glucose in this step. What's important also to note here is this idea of taking carbon from an inorganic molecule and converting it into the carbon that we find in glucose, C6H12O6, is called carbon fixation, and that happens during the Calvin cycle or the dark reactions. In total, this process uses 9 ATP and 6 NADPHs, so it was important that those were made in the light reactions. Okay, once you get to this slide here, you need to pause and recreate this in your notes. Students often will use 2 full pages in their notes, kind of open-faced, but if you can put this in one, you can. Maybe turn it sideways. Be sure to use color, though. That way you can track everything really well. So I'm going to talk about this right now. If you were to kind of zoom in on a leaf or a stem of a plant, so you could look at the stem of this tulip here or a leaf and zoom in, you would see a bunch of cells. These cells on the outside are called epidermal cells, and they surround what are known as guard cells. And these guard cells, depending on how much water they have in them, will either inflate and open or deflate and close. When they open, they create a pore, and these are the stoma or stomata I was talking about, where carbon dioxide can come in and oxygen will leave. Plants also can lose water here. Think about when you walk across the grass in the morning and get your feet wet with dew. That's plants losing water overnight. So carbon dioxide will diffuse into the stomata, and then oxygen will diffuse out. There, you would also see little green dots. These are our chloroplasts. Now, chloroplasts are not just along the guard cells, of course. They're along all kinds of cells along the stem and the leaves, any green plant or any green part of the plant. So we're going to get into all the processes, but I wanted you to kind of get a little bearing for what we're doing here. And then before we get going on the processes, remember what we're doing is we're figuring out where are all of these pieces involved: the water and the carbon dioxide as the reactants, the glucose and the oxygen as the products. So what you're looking at here is a big chloroplast, right? So this little green dot is the same as this chloroplast here. I did not draw it as a double membrane structure, but recall that it is. Inside the chloroplast, I've drawn one of those stacks of thylakoids. It's called a granum. Each thylakoid is green because of the chlorophyll pigment. And I've split it in half so we could separate the light reactions from the dark reactions. But remember that this whole thing is filled with chloroplasts is filled with thylakoids, and the whole thing is filled with syrup or stroma. So in reality, the thylakoid looks more like this down in the bottom right, but we're focusing in on one granum and then the syrup. So I've numbered the steps so we can follow along the process. So photons or light are striking the thylakoids, and we have pigments embedded that are absorbing that sunlight. Water will diffuse in through the roots, will be absorbed by the roots of the plant, and they have special tissues, much like our veins and arteries, that help them transport that water throughout the plant. So the water then is absorbed, and remember, it gets split by the enzymes inside of the electron transport chain. But we take in the water here and the energy from the sunlight, and what comes out of this step, the light reactions, one is the oxygen that then comes out of the stomata and is released to the atmosphere. So if I look over at my plant in the top right, the water comes in and the oxygen will leave actually out to the atmosphere. So the main point of these reactions was not for us. This was for plants so that they could use the energy from the sun and the water to recharge these used up batteries of NADP plus and ADP. They're going to convert these two energetic molecules, recharge them into ATP and NADPH. And just a quick look over in the bottom right, you'll see that ATP, we're going to call that the charged battery. So the adenosine is attached to 3 phosphates, but then when it gets used, we call it ADP. This is like your used battery where one of the phosphates has been cleaved off, but this can get recharged, so it's like a rechargeable battery. Once those batteries have been charged up, they're going to be used in the Calvin cycle, and remember this is happening in the stroma, which is the liquid surrounding all of the grana. So in the Calvin cycle now, carbon dioxide diffuses in through the atmosphere. So looking over at the top right, we have CO2 coming into the atmosphere, being absorbed in through the stoma of the plant, the leaves and the stems. So on the far left, CO2 comes in through those, in between the guard cells, in through the stoma, that the guard cells will create that pore so the CO2 can come in. So CO2 comes in with the help of these two energetic molecules of ATP and NADPH. The carbon from CO2 is now, you'll see I've written carbon fixation here. The CO2, the carbon is going to be taken off of that and then used to slowly build an organic molecule of glucose. So if you look at your overall equation at the top right, this carbon is ultimately going in to this glucose molecule, and we call that carbon fixation, and it happens during the Calvin cycle, and that's very important for you to know. So remember, this is called a dark reaction sometimes. That's because it does not require light directly, but it needed light to build up those energetic molecules. So out of the Calvin cycle comes glucose, and now you might be thinking, well, where does that go? So glucose, if we look at it structurally, molecularly, this is what it looks like. This is going to be remaining inside the plant. It's going to be used to help build structures like leaves, roots, stems, the flowers of plants, fruits of plants. And so it's used in that way, but then also plants can store glucose and use it for their own energy later on. And then subsequent process, we're going to talk about cellular respiration, and plants do that as well. And if you think of a plant, think like a potato or a carrot. All that substance inside of a potato is not for us to consume. It's for the potato itself. When there isn't a lot of energy available from the sun, it can break down all those glucose molecules for energy for itself and get through the rainy season. Okay, be sure you pause the video to get the rest of this slide into your notes. So not all plants follow the process exactly like we just talked about. There are a couple of alternative pathways, a couple of which I'd like you to be familiar with. Now remember that stomata are the pores through which the carbon dioxide enters and oxygen will leave. But water can also be lost here. We call this transpiration. You might remember that from learning about the water cycle. If you're a plant that lives in a hot, dry climate, this is a really bad deal for you. You can dehydrate. When this happens, you're going to increase your oxygen uptake and decrease your carbon dioxide um, intake. And this would actually inhibit carbon fixation. So C3 plants follow the normal process that we just discussed. Remember, you're writing down what's in green. So they follow that normal pathway. But if you're a C4 plant, the stomata will actually partially close when it's really hot, and instead they will make a temporary 4-carbon compound that can enter the Calvin cycle instead of the 3-carbon compound that we saw in the process from before. The plants that do this, I would be familiar with some of the examples, are corn, sugarcane, and crabgrass. This helps them to lose only about half the water that they would otherwise. Cam plants, they actually just open their stomata at night, and they close them during the day. So at night, they take big gulps of carbon dioxide, and they hang on to it, and then during the day, they'll finish up by doing the Calvin cycle. So they will do the light reactions in step 1 so that they can basically create all the ATP and the NADPH they need so that they can do the Calvin cycle during the day. Plants that do this are cactuses and pineapples, and they tend to grow more slowly because of the cooler temperatures, but they do lose the least amount of water. Now, what you're looking at the top right here is not an eyeball of a reptile. Instead, this is an electron microscope image of a stomata. And so here are the guard cells surrounding it. And then this pupil-looking structure is actually the opening, the stomata itself. So here you're looking at some sugarcane. And then bottom right, you're looking at cactuses and pineapples. I know you can imagine corn. Crabgrass is that annoying grass-like plant that grows in your lawn and takes up space. You've probably seen that before. And lastly, we'll end on the factors that affect the rate of photosynthesis. If you understood the rates of enzyme activity, you'll understand this too. It's basically related because of enzyme activity. As we increase light, it increases the rate of photosynthesis, as you see in graph A, but ultimately it can only speed up so much. When the light intensity continues to increase, essentially the enzymes are doing all that they can and they become saturated with the substrate and it simply can't go any faster. Now, the same graph would occur from carbon dioxide levels. The more carbon dioxide we have, the faster the rate of photosynthesis to a point. At some point, all of the rubisco is taken up and cannot bind any more carbon dioxide, and so it will level off. Then lastly, if we keep increasing the temperature, this will speed up the rate of photosynthesis to a point, but then it becomes too hot for the enzymes, and one of two things will happen. If it continues to heat up, it could actually denature the enzymes. Remember, they're made of proteins, so the active sites will become denatured. They will change shape, and they can no longer bind their substrates, and so the photosynthesis would slow down. But usually what happens is when it becomes too hot, plants will actually close their stomata and just as a self-protective mechanism, and they will slow down their photosynthesis. So I hope that helps. Make sure you've gotten everything that is in green in your notebooks, and we'll see you soon.